Chapter 4, Segment 1. What and where pathways? So cells in the V1 are interested in basic features of the visual image, responding to edges or lines of specific orientation, motion, and size. A cell will respond to its preferred stimulus only if that stimulus is presented in a very specific location relative to the point where the observer is fixating on its gaze. So the extrastriate cortex is a set of visual areas, so called because they lie just outside the primary visual or striate cortex. From the extrastriate region of the occipital lobe of the brain, visual information moves out along two main pathways. One pathway heads up to the parietal lobe. The visual areas in this pathway seem to be important for processing information relative to the location of objects in space and the actions required to interact with them. So these are sometimes known as the where pathways. And then other pathway heads down to the temporal lobe and is known as the what pathway. And this one appears to be the locus for the explicit acts of object recognition. As the name implies, what is in view seems more important than where it is, but some basic object information represented in both pathways, and some where information is encoded in the temporal lobe to the what pathway. Evidence for a relationship between the temporal lobe and object recognition come from studies where large sections of the temporal lobe were lesioned in monkeys, and these monkeys behaved as though they could see but did not know what they were seeing. This is known as agnosia. The inferotemporal cortex is particularly important in the visual problems of these monkeys. So Charlie Gross began to record the activity of single cells in this area. So cells in the inferotemporal cortex were discovered to have receptive fields that could spread over half or more of the field of view. And spots and lines didn't work as well, but the silhouette of a monkey's hand worked wonders. And the silhouette of monkeys' faces excited other cells. So Horace Barlow suggested hierarchical model of visual perception in which the small receptive fields and simple features of visual cortex are combined with ever greater complexity as one moves from the striate cortex to the IT cortex. So the term grandmother of the cell refers to any cell that seems to be selectively responsive to one specific object. Recent research casts doubt that IT cells respond to an object independent of its position in the visual field. It does seem clear that cells in that part of the brain are critically involved in object recognition. The IT cortex maintains close connections with parts of the brain involved in memory formation. IT cells need to learn their receptive field properties. So Nikos Logothetis demonstrated that IT cortex have plasticity, and he trained monkeys to recognize novel objects and found IT neurons that responded with high firing rates to those objects, but only when the objects were seen from viewpoints similar to those from which they had learned. So human cortex has areas that appear to be the equivalent of the monkey IT cortex. These are known as homologous regions, when they have the same function but different species. So Quiroga et al. Made recordings from single cells in the temporal lobe of human observers. The observer looked at the collection of images while the activity of a cell was monitored. Some cells turned out to have specific tastes. We have a growing volume of functional imaging data that document areas in the human brain that appear to be specialized for different sorts of stimuli. And these tend to be simply named, like the fusiform face area is interested in faces, and the parahippocampal place area responds to spaces in the world like rooms with furniture. Humans with lesions in the temporal lobe often show signs of agnosia. An agnosia is the ability to see without the ability to know what is being seen, and this can sometimes be specific. So processing that leads to the categorization of objects and scenes can be very fast. If a picture is flashed to an observer and asked whether it contains an animal, a signal can be recorded in the observer that reliably differentiates animal from non-animal scenes within 150 milliseconds from the onset of the stimulus. This is fast enough that it cannot be a lot of feedback from the higher visual or memory process.